Well, if we look at the method from start to finish, we have getting the correct piece of DNA, inserting the DNA into a plasmid, and finally getting the plasmid into the cells. Sounds simple. Well, let's assume that getting the correct piece of DNA is not an issue. The next step is inserting it into the plasmid. We use a restriction enzyme to cut the plasmid and the piece of DNA we wish to insert. And this is where we can have some problems. First, does the plasmid contain a restriction enzyme site that we can use? Second, if we use just one restriction enzyme, then the plasmid can ligate back upon itself and contain no insert. This is true if we use a restriction enzyme that produces blunt or sticky ends. This is obviously no good, as we won't have cloned the DNA. The plasmid will have been repaired and have no insert. Plus, if we're trying to express the protein, then there is no guarantee that the insert is in the correct way around, with the reading frame aligned with the expression machinery of the plasmid. You can see this orientation problem in this demonstration. Say we cut with an enzyme called EcoRI. Then the insert can go in either orientation. You can see how it can flip around and bind in either way. Plus, the plasmid could ligate back on itself as it has the ends that match. If we cut the plasmid using two enzymes, say EcoRI and HindIII, then the plasmid cannot ligate to itself and the insert can only go in one orientation, as in the other orientation the ends don't match. So the best option is to use two restriction enzymes that produce two different sticky ends in the plasmid and the insert. Next, getting the plasmid into the cells. How do we know a bacteria has taken up the plasmid? And how do you know that the plasmid contains the insert? A plasmid is just a circular piece of DNA which can be anything from 1,000 to 200,000 base pairs in size. Although most of the ones I've worked with in the labs over the years have been 4,000 to 6,000 base pairs. All plasmids have one thing in common, and that is they all have an origin of replication. If they didn't, then copies couldn't be made in the cell. Although plasmids occur in nature, the plasmids which we use in the lab have been engineered to make cloning easier. For example, they will contain a multiple cloning site, which will have numerous restriction enzyme cut sites, and this makes cloning in a piece of DNA a lot easier for scientists, as it gives us more options to the restriction enzymes that we can use. There is a possibility that the bacteria may not take up any of the plasmids. Therefore, to help us with this problem, plasmids have been engineered to contain an antibiotic resistance gene. And if the plasmid is used with a bacteria that is sensitive to the antibiotic, then only bacteria that have taken up the plasmid and therefore now contain the antibiotic resistance gene will grow on media that contains that antibiotic. This means we can select for bacteria that have been successfully transformed, that is, bacteria that have taken up the plasmid. Now, just because the bacteria contains the plasmid, it doesn't mean that the plasmid contains the insert. One of the methods we use in the lab to help select for bacteria that contain the insert is a double selection method, and it is often referred to as the blue-white selection method. The blue-white selection method uses antibiotic resistance to select for the bacteria that have taken up the plasmid. And the multiple cloning site is engineered so that if it contains an insert, it will disrupt a gene that has been previously cloned into the plasmid. The gene that is used is called LAC-Z, and it's from the lac operon, and it makes an enzyme that can convert a chemical called XGAL, which is an analogue of lactose, to give a blue colour on the plate. Therefore, if the DNA insert has been successfully cloned into the plasmid, then the LAC-Z gene will be disrupted, which means that the bacteria can no longer metabolize the XGAL to make a blue-white colony. Therefore, the colonies appear white. Hence, we have a way of selecting for bacteria that contain the plasmid and also contain the insert. That is, they produce colonies that are white and not blue. In addition to the characteristics I have talked about earlier, a plasmid may also contain sequences and systems that allow the DNA insert to be expressed. And in some cases, the plasmid will contain code that allows the protein to be tagged to help with purification. Now, the type of plasmid we have been talking about does have some limitations in terms of the size of the insert it can handle. Once the size of the insert gets above 10,000 base pairs, it can no longer fit in the insert site, as the insert is now typically bigger than the plasmid. Therefore, in the lab we use vectors like bacterial artificial chromosomes, or BACs, which can accept DNA inserts up to 300,000 base pairs, or we can use yeast artificial chromosomes, YACs, which we use with yeast, and can accept inserts up to 1 million base pairs. In this video, we've looked at how we can overcome some of the potential problems we face in the lab when we try to clone DNA into a plasmid.